micro students, welcome to my lecture on chapter 10. In chapter 10 we're going to be talking all about genetic engineering, and we will cover some of the tools and techniques and how those can be applied. We're also going to cover some of the features of DNA that lend DNA to these techniques, and then we're going to talk about some of the applications, both medical and otherwise practical applications of genetic engineering. In general, when it comes to genetic engineering, scientists' basic knowledge about DNA is used to come up with techniques that can be used for practical purposes. That's known as applied science. It's also used to make products for various industries. Bioengineering is directly and deliberately modifying an organism's genome for either research or practical purposes. The field of biotechnology is using an organism's biochemical and metabolic pathways in order to produce something or some things that are useful for industrial production. Okay, so some of the tools and techniques of genetic engineering include the basic knowledge that if you heat DNA to 90 degrees Celsius to 95 degrees Celsius, it will cause the two strands to separate. Nucleotides can be identified this way. They can also be replicated or transcribed. When the DNA is then allowed to cool slowly, then the complementary nucleotides will reform the hydrogen bonds and DNA will become double-stranded once again. In summary, heating DNA causes it to denature, which breaks the hydrogen bonds between the bases in the complementary strands. And since DNA rejoins and reforms those hydrogen bonds between the complementary strands when it is allowed to cool slowly, it allows for all kinds of things. For one, it allows DNA from different organisms to be put together into complementary strands as long as they have some matching sequences. Scientists have also figured out how to use enzymes from organisms in order to cut and put back together DNA, as well as to make DNA out of mRNA templates. Restriction endonucleases are enzymes that recognize specific sequences in DNA and actually cut the DNA into shorter fragments. Those can be useful to, obtain shorter sequences or segments of DNA that can be more easily managed in the laboratory for research or practical purposes. Those restriction endonucleases actually break the phosphodiester bonds between nucleotides in a strand of DNA. They can be used to cut DNA at specific sites, such as palindromes. A palindrome in DNA is just like a palindrome in English. It's a sequence that reads the same 5' to 3' as it does 3' to 5'. When restriction endonucleases are used to cut DNA into smaller fragments, those are called restriction fragments, and even in the same species, a particular restriction endonuclease can cut the DNA up into different fragment lengths, and that RFLP means restriction fragment length polymorphisms, and that can allow for scientists to compare the cutting patterns of DNA from two different organisms with the same endonuclease based on the differences in the restriction fragment lengths that produces, which is one of the steps in preparing genes from different organisms to be spliced or joined together. Ligase is an enzyme that we talked about when we talked about DNA replication. It is an enzyme that is used to rejoin small fragments of DNA, and it can be used to splice genes into plasmids and other chromosomes so that you can combine, or scientists can combine DNA from different organisms. Reverse transcriptase is an enzyme that can make a DNA copy from an mRNA, tRNA, or rRNA, so messenger, transfer, or ribosomal RNA template, and it allows scientists to synthesize or make eukaryotic genes from mRNA transcripts, which do not have the introns, the parts that aren't coding. And this is a figure showing that use of reverse transcriptase. And so, you know, just as a little refresher, you have your DNA that has the exons, which are expressed or code for amino acids, and the introns, which are spliced out. And so you have your DNA, it's transcribed into mRNA. This is the original strand that's transcribed. Then the splicing removes the intron sections, and here's your finished mRNA transcript. Well, then you can apply the reverse transcriptase to the mRNA transcript and end up with a strand of DNA that does not include those introns. And then you can use DNA polymerase to make the complementary strand to the strand that was made using the reverse transcriptase. Then these double-stranded DNA sequences that have been made using this process can be readily translated or used to create proteins by bacteria. And this type of DNA, the cDNA, complementary DNA that's produced through this process, can also be a really good tool that scientists can use for converting RNA for sequencing, analyzing ribosomes, and also for identifying RNA viruses. And the restriction fragments produced by these endonucleases can be analyzed based on length or size through a technique called gel electrophoresis or DNA gel electrophoresis, which some of you may have used in labs, either in a general biology class in high school or in college. And so what you do is you cut the DNA with an endonuclease or endonucleases, and then you use an agar gel. There's a gel in this contraption, and you make wells in the gel when you're molding the gel, and then you can put the DNA samples in those wells and use an electric current to pull the DNA fragments through, or the DNA samples. These are the wells. You can kind of see the samples loaded into there, and then there's loading dye that makes it visible, and then the electric current pulls the DNA from the negative end to the positive end and separates it by size. The gel itself has holes or pores in it, and so the smaller fragments move farther down the gel, and the bigger ones or longer ones don't make it as far down, so it actually separates them in space based on the length of the fragment. Okay, so since DNA has a negative charge, that's why it is pulled to the red or the positive end of your DNA gel electrophoresis apparatus. And like I said, the rate of movement depends on the fragment size. Larger fragments move more slowly, and smaller ones move more quickly through those pores. And so what you end up with, so this is your original where you load your sample into here, and as you run that current through it, it starts pulling the DNA down the gel. And then what you end up with is you end up with these bands. So these are each different samples of DNA from a different source, or the same DNA cut with different endonucleases. And so you end up with this different banding pattern, meaning that you have different fragment lengths. And that brings us back to that restriction fragment length polymorphism. And that's why you end up with these banding patterns, because of the different sizes of fragments produced. Here's a picture of what a gel would look like. And so then you can actually relate the banding pattern to the number of base pairs that each band has in it, aka the length. So this one has 23,130, which is the most, and then 9,416, 6,557, 4,361, all the way down to 2,027. And then you have some obviously down here that are going to be even shorter. Okay, so other ways to analyze DNA: nucleic acid hybridization, which unites single-stranded DNA with a complementary strand of either single-stranded DNA or RNA, or you can cause RNA to hybridize or join with another RNA strand. So nucleic acid hybridization is a technique that can be used to create gene probes, and it's just using short fragments of DNA that have a known base pair sequence that will basically catch or help to find complementary stretches of DNA if there's one in the sample. So if you have a gene probe that is A, T, C, G, C, then it's going to find stretches of DNA in the sample that are complementary, and so there'll be T, A, G, C, G. The Southern blot method is a technique that combines DNA gel electrophoresis with gene probes, and you have the DNA fragments separated by electrophoresis. They're then denatured so that they become single-stranded, or the strands separate, and then they are incubated with the DNA probes, and the probes attach to complementary segments in that DNA if it's present. Another technique that's used is called FISH, or fluorescent in situ hybridization, and that is the use of probes to try to find specific sequences within intact cells. And so you can use that for diagnostics or locating like medical diagnostics, or locating genes on a chromosome. In FISH, the probes are labeled with fluorescence, and when they're placed either into an intact cell or incubated with DNA, then they're looked at under a UV microscope, and you can actually see the fluorescence, which shows the locations of particular genes. I just wanted to point out that there is this slide in your slideshow. It's slide number 22 in the PowerPoint that's in the course shell, and it just has a list, and it's in section 10.1 in your ebook, and it just has a list of these different research fields in the study of genomics. So I just wanted to point that out. Last section of 10.1 has to do with DNA sizing, sequencing, and synthesizing. DNA sequencing is determining the order of nucleotides or bases in a strand of DNA, and can be applied to all types of DNA from different organisms. Frederick Sanger invented the technique. The most common way of doing this is called the Sanger technique. And so you get test strands, usually strands that have been cut with endonucleases to be manageable in the lab. They're heated to make the strands, the complementary strands, come apart, and so that they can be used as a template to create complementary strands. The fragments are divided into tubes that include the materials needed to synthesize DNA. The primers, which tells the DNA polymerase to add nucleotides to the 3' end, the DNA polymerase itself, which is responsible for elongating the strand, and then the 4 nucleotides, A, T, C, and G. In addition, fluorescently labeled dideoxynucleotides are added, and those are used to actually figure out the sequence of the DNA. They're missing a hydroxyl group, and so when they bond to a point in the strand of nucleotides, it causes that to stop elongating, and since they're fluorescently labeled and they will randomly bond to the end of DNA stretches, you end up being able to use the color pattern formed in order to actually figure out the sequence and the order of the DNA. So that's the Sanger technique. I'm going to do 2 more lectures and get through the rest of this chapter. Thank you for listening to part 1.